This video was made by both the BUCT&ZJU China team from Zhejiang University and the BUCT&ZJU team from Beijing University of Chemical Technology. Today, we are going to introduce gene editing technology from 4 aspects: background, 3 main methods, applications, and a video for understanding. Due to time constraints, we may publish the 4th part later. With the rapid development of molecular biology in the 21st century, genetic engineering has become a new research hot issue based on the original molecular genetics. When it comes to genetic engineering, we must pay attention to gene editing technology, which has gained widespread attention recently. Among them, the CRISPR-Cas9 technology, known as the molecular scissors, won the 2020 Nobel Prize in Chemistry. This genetic manipulation can modify the target gene at the genomic level to achieve knockout, addition, mutation of specific DNA fragments, and to perform precise surgery on the genome. The gene editing system includes an RNA guide, DNA recognition model, and an RNA-guided DNA cleavage or modification model. The introduction of foreign genes into the recipient genome depends on DNA repair mechanisms, the most important of which is non-homologous end joining. Because this mechanism does not require any template, the repair protein directly pushes 2 broken DNA strands together and rejoins them with the help of DNA ligases. Moreover, the activity is relatively high, but it is more prone to errors. The NHEJ can repair broken DNA at all stages of the cell cycle. NHEJ is also involved in the regulation of diversity, antibodies, and T-cell receptors in the immune system. The DSB, double-strand break, is first recognized and bound by the Q70-Q80 complex, then binds to DNA-dependent protein kinase, DNA-PK, to form the DNA-PK complex. The NHEJ initiation complex DNA-PK subsequently binds to the broken DNA ends. Then, it recruits subsequent NHEJ repair factors, such as XRCC4 and DNA ligase IV, to repair the broken DNA. The core design idea of CRISPR editing technologies is to find a specific sequence through the recognition model, use the functional model for incision digestion, and then use the DNA repair mechanism to achieve gene knockout, insertion, mutation, and other editing purposes. Nowadays, gene editing technology has advanced through 3 generations, including ZFNs, TALENs, and CRISPR-Cas. The ZFN and TALEN complexes recognize the triplex-based motif through their zinc finger or tail domains and cleave the recognition sequence. CRISPR-Cas9, on the other hand, recognizes DNA base pairs with the help of guide RNA and also attaches a nuclease for DNA cleavage. Both technologies are based on a protein DNA recognition model, combined with non-specific nucleases to function. CRISPR stands for clustered regularly interspaced short palindromic repeats, and it is a system that is a whole protein family, including many proteins that achieve the CRISPR function. The most important part is the Cas9 protein because there are 2 in the nucleus size, and only this mechanism exists in bacteria and microorganisms similar to the human immune system. After the phage invades bacteria, the genetic material will be integrated into the bacterial genomic DNA. When the phage of the same kind invades again, the integrated fragments in the protein are then transcribed into RNA and linked with a functional tracer on a former guide RNA, which will then bind to the Cas9 protein. The invasion sequence is basically recognized by the guide RNA, and then the Cas9 protein is used to cut the nuclease, so that the phage loses its infectiveness. After the efforts of generations of scientists, Jennifer Doudna and Emmanuel Charpentier published an article in Science in 2012 to prove that artificially designed guide RNA can guide Cas9 to achieve site-specific cleavage. Laurent and Jennifer received the 2020 Nobel Prize in Chemistry. In 2013, the team of Chinese-American scientists Feng Zhang and George Church applied CRISPR-Cas9 gene editing technology to mammalian and human cells. Since then, CRISPR-Cas9 gene editing technology has been widely used through 2 specific recognition bases: pairing of guide RNA and target sequence, and binding of Cas9 protein complex, and show DNA sequence for TOS-pacer adjacent motifs compared with protein. The base pairing of RNA is more specific and stable, making this technology full of application prospects. Gene editing technology has a wide range of applications. In 2022, Gao Kaiksha, the The Chinese Academy of Sciences and the QRU Jinlong's team used CRISPR-Cas9 technology to achieve directional mutation and obtain varieties with broad spectrum, powdering resistance, all affected growth, and promising higher yields. In 2022, the U.S. Food and Drug Administration approved the first gene-edited beef cattle product to enter the market, believing that it features lower safety risks. Of course, hope is often accompanied by anxiety. Examples such as gene-edited babies may also consider the boundaries of technology. The accuracy and safety of gene editing are often key factors in people's acceptance of gene editing technology and the subsequent ethical issues of gene therapy. Therefore, it is necessary to improve laws and regulations to further standardize the application of technology in order to fully leverage its advantages.